Bismillah ar-Rahman ar-Rahim. Assalamu alaikum everybody. In our last presentation we started talking about receptors as a major site of drug action. We said that receptors, they might be membrane receptors, cytosolic receptors, or nuclear receptors. We said that most drugs, they act on membrane receptors, and we started talking about membrane receptors. We said that they belong to three major families. First are the ion-linked receptors. The second are the enzyme-linked receptors. And the third are the Gproteinlinked receptors. We started talking about the ion channel linked receptors. We said that when the agonist bind to these receptors, this will lead to opening of certain ion channels, which will change permeability to certain ions, and therefore will change the excitability of the membrane towards either more excitation, and we have taken an example of nicotinic receptors stimulated by acetylcholine on the neuromuscular junction, or towards inhibition, and we have taken example of GABA on GABA-A receptors. Today, inshallah, we'll finish talking about the different types of receptors. First, we'll talk about the enzyme-linked receptors, and then we'll talk about the Gprotein-linked receptors. So these are the main goals of today's lectures, is to know what are the different types of enzyme-linked receptors and what are the functions, in brief of course. Then we'll talk about the Gprotein-linked receptors, what are the type of receptors linked to Gprotein, what are the types of Gprotein, how they are activated, and how they are linked to ion channels and membrane enzymes. So, first we talk about the enzyme-linked receptors. These are a single polypeptide chain, which is a protein, of course, like any other receptor. They are protein in nature, composed of a single polypeptide chain which crosses the membrane, biological membrane. It is composed of an extracellular component. This extracellular component or extracellular domain contains the binding site for the agonist, and an intracellular domain which is the enzyme. That's why they are known as enzyme-linked receptor. Therefore, the composition of these type of receptors, they are an extracellular domain which contains the binding site, and an intracellular domain which contains the enzyme or which has the catalytic function. Enzyme-linked receptors, they are of three major types. First, which is the most known and most famous, are the tyrosine kinase linked receptors. We'll talk about them shortly. Second are the serine thionine receptor kinases. Third are the guanylyl cyclase. Actually, guanylyl cyclase, some people, they divide it into the cytoplasmic receptor. So tyrosine kinase is the most famous of the enzyme-linked receptor. So what will happen? Let us take an example of insulin. Insulin, all we know about insulin, which is secreted by the beta cells of the pancreas. Insulin, what it is secreted, it will act on a specific insulin receptor present on the extracellular ligand binding site. It will stimulate tyrosine kinase, and this stimulation of tyrosine kinase will stimulate many intracellular enzymes, which ends up in the actions of insulin, including, of course, different actions, including uptake of glucose, uptake of amino acids, stimulation of other cytoplasmic enzymes responsible for the carbohydrate metabolism, for the lipid metabolism, and so on. Okay, this is an example. Tyrosine kinase nowadays, they are very famous. Why? Because they have found that many of the cancer cells, most of the cancer cells show very high activity of tyrosine kinase. That's why now there are many drugs which are used in treatment of cancer. They act by inhibition of tyrosine kinase. There are tyrosine kinase inhibitors that we'll discuss inshallah in details when we talk about oncogenesis and their treatment. Okay, so this is the first time enzyme-linked receptor, and these enzyme-linked receptors, they are of, as we have said, it is a single polypeptide protein which crosses the brain just one time with an extracellular domain containing the binding site and intracellular domain which contains the enzyme, and the most famous is tyrosine kinase enzyme, and many hormones, as we will see, and many neurotransmitters, they act through the enzyme-linked receptors. This is the first type. A second type, the third type of receptors are the receptors which are linked to G-protein or Gprotein-linked receptors. Why they are named G-proteins? Why? Because they have the ability to bind with guanosine triphosphate and guanosine monophosphate. They have binding sites for guanosine triphosphate and guanosine monophosphate. That's why they are known as G-protein. So what is the main function of the G-proteins? What they do? Actually, they act as a mediator between receptor and ion channel and receptor and membrane enzymes. So they act as a mediator between receptor and ion channel and the receptor and membrane enzymes. So if we just look at the system as a whole, so this is the receptor. We'll talk about them in details in short minutes. And this the G-protein, and the G-protein acts as a mediator between the receptor and ion channel or membrane enzymes, which are an OR effector. So our system is composed of three components. First are the receptors, second are the G-proteins, third are the effectors, which are either ion channels or membrane enzymes. This process starts with the stimulation of the receptor by an agonist, and stimulation of the receptor will activate the G-protein. Once the G-protein are activated, they lead to opening of ion channel, and as we know, opening of ion channel will change the membrane excitability, or they will activate other cellular enzyme, and this activation of cellular enzyme will produce different intracellular effect by what's known as second messenger. So we have first messenger and we have second messenger. Okay, therefore the G-proteins, they act as a mediator between receptors and ion channels and membrane enzymes. So first we'll talk about the property of the receptors which are linked to G-protein, because they have a specific property different from other types of receptors. Then we'll talk about the G-proteins, how they look like, what is their structure, and how they are activated. Then we'll talk about how this G-protein will affect ion channels or will affect membrane enzymes. Okay, so first we'll talk about what are what is the main structure of receptor linked to G protein? Actually, the receptor which are linked to G protein, they are a single polypeptide chain which crosses the membrane 7 times, as you can see. This is a polypeptide chain, single polypeptide chain which crosses the membrane 7 times. As you can see, this is first time, second, third, fourth, fifth, sixth, seventh. Okay? It is single polypeptide protein with an extracellular terminal, which is the amino group, and intracellular terminal, which is a carboxyl group, carboxy group. Okay? In the extracellular N-terminus, there is the binding site for the agonist. Actually, most of the agonists either they bind on the extracellular N-terminal or in pockets present near the extracellular N-terminal. The intracellular C-terminal contains the binding site for G-protein, contains the binding site of G-protein. Okay? So, Gprotein coupled receptors, they are known as Gprotein coupled receptors, and also they are known as 7 transmembrane receptors. They are known as 7 transmembrane receptors. Transmembrane receptors, or 7TM. Of course, they have very different structures and they belong to the very different families, but they share the property that they are of one single polypeptide which crosses the membrane 7 times with intracellular and extracellular terminus. Okay? We have to know that Gprotein coupled receptors, they are the most important type of receptor present in the body because, as we will see, they control most of the important physiological function. For example, the function of the autonomic nervous system, whether it is adrenergic or cholinergic, most of the receptors are of the Gprotein coupled receptors. Function of cardiovascular system, function of the autonomic nervous system, many of the central functions that belong to the Gprotein coupled receptors. Second, in about 50% of the drugs which are used in clinical use, they act through the Gprotein coupled receptors. Okay? So this is first the structure of the receptor which are coupled to Gprotein. There are 7 transmembrane single polypeptide chain with an extracellular domain which has the agonist binding sites or an antagonist, it depends, and an intracellular domain which has a Gprotein binding site. Okay, this is first. And second, we will know what's the structure of Gprotein. What's the structure of Gprotein? Gprotein structure. Gprotein is heterotrimer. What does it mean, heterotrimer? Hetero means they have a different structure. Trimer means that they are formed of 3 protein subunits. Hetero means they are of different structure, as you will see, and trimers means that they are of 3 subunits. They are formed from 3 subunits. So how they look? This is a draw of how they look. So Gprotein is composed of 3 subunits: G alpha subunit, G beta subunit, and G gamma subunit. These are the trimers. G alpha, G beta, and G gamma. Okay. G alpha subunit is the most important. Why? Because G alpha subunit is most important because it contains the binding site for guanine nucleotide for the GDB and GTB. G alpha subunit contains the binding site for the guanine nucleotide, whether it is GDB, guanosine diphosphate, or guanosine triphosphate. Also, it contains the intrinsic GTPase activity. The intrinsic GTPase activity, which breaks down GTPase, or which breaks down GTP after ending the action. The alpha subunit also determines what is the type. We said that they are hetero. Hetero means they have a different protein structure, and which determines the protein structure is the alpha subunit. So alpha subunit, up to now there are 17 different types alpha subunits which are discovered in a human body, and the widely spread, the most known are the GS alpha or GO alpha or GKQ alpha. Okay, therefore the alpha unit of the G protein, it contains the binding site for guanine nucleotide. It has the intrinsic GTPase activity for the breakdown of GTP after finishing the action, and it is of diverse structure and determines the type of the G protein. G beta or gamma, usually they act as a dimer. They never separate. They stay as they are. They act as a dimer. When they are activated, they act together, as we will see later in a few minutes. Okay. So this is the general structure of the G protein. As you can see, this is the receptor, a 7-transmembrane receptor, which was composed of 7 of a single polypeptide, which crosses the membrane 7 times with extracellular domain and intracellular domain for the binding of G protein. And this is the G protein, which is usually floating into the inner part of the cell membrane. And this is the alpha subunit, and this is the beta and gamma subunit. So how G protein is activated? How G protein is activated? We have said last lecture about the activation of the receptor, how receptors are activated by orthosteric binding or allosteric binding site. Now we'll talk about how G protein is activated. As you can see, when the receptors are not stimulated, when there is no agonist, the G protein is present inactive as a trimer. The alpha, beta, and gamma subunit, they are present as one unit. Once the agonist binds to the receptor, it will change the configuration of the receptor, i.e. the receptor will be activated. It will attract the G protein molecules towards the receptor. In this moment that the G protein binds to the receptor, in this moment the exchange of GDP for GTP. GTP will enter the G alpha subunit and GDP will leave the subunit. At this moment, the G protein will become activated. In that case, it will separate into two active subunits. Activated alpha subunit and activated beta and gamma complex. Two subunits that are active. These active alpha subunit or beta and gamma complex subunits will either activate membrane ion channels, as we will see later, because they are rich in energy. What they do, they give energy to ion channels to open. Also, they give energy, or they phosphorylate membrane enzymes, and these phosphorylation membrane enzymes will lead to formation of what are known as second messenger. Okay. Now, so activation of G protein is done when the agonist bind to the receptor. Once the agonist binds to the receptor, will attract the trimer and to bind to the intracellular domain, and once it binds to the receptor يحصل exchange of GDP with GTP and separation of the trimer into two dimers, one activated alpha subunit and the other beta and gamma complex. These two, they are rich in energy. They will activate either ion channels, changing the excitability of the membrane, or they activate membrane enzymes forming second messenger. So this is what we'll talk about in the next few minutes. First, how membrane ions are activated by the G protein, or how the membrane enzymes are activated by the G protein. We have to say here that once the receptor is activated, it can activate about 100 molecules of Gprotein. That means the signal is amplified. Amplification of the signal takes place. That means one agonist binding to one receptor, one agonist molecule binds to one receptor, it can activate 100 molecules of G protein. Therefore, a signal will be amplified. The signal will be enlarged. As we will see later. Okay. So first we'll talk about how membrane ion channels are activated by G protein, and then we'll talk about membrane enzymes and their activation by the G protein. So first, we'll talk about G-protein gated ion channel. Let us take example to make it easier for us to understand. Example is the muscarinic receptor type 2, M2 receptors, muscarinic receptor, which are present on the cardiac muscle, and the G protein is the GI protein. GI is the indication of inhibition, means that stimulation of this G protein will result in inhibition of activity, and the ion channel are potassium channels. So what will happen in this case? Suppose this is the muscarinic receptor type 2, which are present on the cardiac cell, on the myocyte. Once the agonist bind to the receptor, it will change the configuration of the receptor in a way that it will attract the G protein, and the G protein will be activated. And once it is activated, to split into alpha subunit and beta and gamma subunit. The beta and gamma subunit, once it is activated, it will activate potassium channel. It will lead to opening of potassium channel. As we know, potassium is concentrated inside. It will rush outside of the cytoplasm. Once it rushes outside, it causes membrane hyperpolarization. This we have explained last lecture. Membrane hyperpolarization. Membrane hyperpolarization means the membrane is inhibited, therefore decreasing the excitability of the membrane, and this will lead to inhibition. So just imagine that this takes place in the SA node of the heart. SA node, as we know, is the pacemaker of the heart, that it controls heartbeat. So in this case, if the SA node is hyperpolarized, that means decrease in activity. That means decrease in the activity of SA node. That means decrease in heart rate. decrease the heart rate. This is bradycardia. Okay, yep, this is very famous example of G-protein gated ion channel. And second, we said that Gprotein acts as a mediator between either ion channels, between a receptor and ion channel, or between a receptor and membrane enzymes. So this is an example of how Gprotein acts as a mediator between a receptor, and we have taken the example of receptor muscarinic receptor type 2, and ion channel, which are the potassium ion channels. Second, how the Gprotein acts as a mediator between a receptor and membrane enzymes. How the Gproteins, after their activation, will activate membrane enzymes. We will talk about two very important enzymes. The first is known as adenylyl cyclase, the second is known as inositol phosphate system. So we'll talk first about adenylyl cyclase. Adenylyl cyclase, we'll take an example of receptor, which are the beta 1 receptor and beta 2 receptors. Beta 1 present in the heart myocytes, or beta 2 present in the smooth muscles of the lungs or the uterus, it depends. And Gprotein is the GS, as we said that Gproteins, they are hetero, they have different structure, and in this case the type of protein GS indication of stimulation. That means the activation of Gprotein will result in stimulation of activity or inhibition. It depends on the enzyme affected. And the membrane enzyme is adenylyl cyclase. Adenylyl cyclase. So what will happen in this case? Let us see our system. Once an agonist binds to the system, binds to the receptor which are present on the cell membrane, it will attract the Gprotein, and Gprotein will split into two units, as we have said, G alpha and G beta and gamma. The G alpha subunit would activate a membrane enzyme known as adenylyl cyclase. This is a membrane enzyme. Membrane enzyme موجودة in the membrane. Once the adenylyl cyclase is activated, which is the membrane enzyme, it will convert adenosine triphosphate into cyclic AMP. This is adenosine triphosphate. Adenosine triphosphate. Okay, it will convert adenosine triphosphate into cyclic adenosine monophosphate. This is monophosphate. This is the second messenger. Why it is known as second messenger? Because it is responsible, its formation will result in intracellular action. That's why it is known as second messenger. Second messenger. Okay? Why? Because the first messenger is the agonist, which binds to the receptor. This was the first messenger. First messenger, the agonist which binds to the receptor. The second messengers are formed after activation of certain membrane enzymes. Cyclic AMP, once it is formed, it is rich in energy. Now, it will lead to activation of other cellular enzyme known as protein kinase A. Protein, as we can see, there are different types of protein kinases. This cycle can be specific in activating protein kinase A. Protein kinase A. So, protein kinase A, it was inactive in the cytoplasm. By the cyclic AMP, which is the second messenger, it is converted to active protein kinase A. This protein kinase A, once it becomes active, it would either stimulate other cellular enzyme or inhibit other cellular enzyme. You see? So protein kinase either produce stimulation of other cellular enzyme or inhibition of other cellular enzyme. Let us take example of stimulation and inhibition. First, in case of stimulation, once an active protein kinase it becomes active, it will lead to phosphorylation of the contractile protein troponin 1, which is a contractile protein in the cardiac muscle, and this will result in contraction of the cardiac muscle. Okay? Therefore, active protein kinase A will lead to phosphorylation of the contractile proteins troponin 1 in the cardiac muscle, and this will result in cardiac muscle contraction. Therefore, we can say that stimulation of beta-1 receptor in the heart can increase cardiac contraction through phosphorylation of the contractile protein troponin 1. Okay? A second example is of inhibition, in which active protein kinase A can phosphorylate myosin light chain kinase, which will lead to myosin light chain kinase, which will lead to inhibition of myosin function. And we know that myosin light chain kinase is the responsible for the providing energy for myosin to slide over actin. Therefore, the further phosphorylation of the myosin light chain kinase will inhibit myosin function. That means relaxation of the smooth muscles of the bronchioles. Okay? Okay. Now, so again, we said that adenylyl cyclase, an enzyme which is activated by Gprotein, that will result in formation of second messenger cyclic AMP. Cyclic AMP will activate protein kinase A, which in its turn either activates cellular enzyme like troponin 1, resulting in cardiac muscle contraction, or it leads to further more phosphorylation of myosin light chain kinase, which will inhibit myosin function, and this will result in smooth muscle relaxation. Okay, this is first very famous example. We'll take many drugs which will act through this system, as we will see later, mostly with the adrenergic drugs and many hormones. So first, Gprotein activated membrane enzyme is through the adenylyl cyclase system. A second system through phospholipase C-B enzyme, which is known as phosphatidyl inositol system. Phosphatidyl inositol system. This is a very complicated system, which I will try to explain easily. It is involved in many cellular functions and very important functions as well. So phospholipase C, there are different isoforms of phospholipase C. There is phospholipase C, B, P, and there is phospholipase C, gamma, and so on. But in this case, we are talking about phospholipase C, beta isomer. Once an agonist like, for example, alpha-1 agonist binds to the alpha-1 receptor, it will lead to stimulation or change in configuration of the receptor in a way that it would attract Gprotein. Once Gprotein is activated, it will, as we said, divide into two isomers, one G alpha unit and one G beta unit. So once Gprotein is activated, as we have said, exchange of GDP instead of GTP, And this activated G protein, it will activate phospholipase C beta, which is present in the, it is phospholipid, it is present on the membrane. Phospholipase C, once it is activated, it will act on what's known as phosphatidylinositol biphosphate, or IP2 phosphatidylinositol diphosphate, and convert it into 2-second messenger. Converted into 2-second messenger. One is known as diacylglycerol, or DAG, and the other is known as inositol triphosphate, or IP3. Okay? Okay, now, so once the agonist binds to the receptor, it will activate Gprotein. Once the Gprotein is activated, especially alpha subunit Gprotein, alpha subunit will activate phospholipase C, which is a membrane enzyme. Phospholipase C will act on a membrane phospholipid known as phosphatidylinositol diphosphate, or biphosphate. This will be converted into 2-second messenger, not one, 2. Diacylglycerol, which is known as DAG, and inositol triphosphate, or IP3, okay? 2-second messenger. A DAG will stay floating in the plasma membrane. We'll talk about it in a few minutes. IP3 will diffuse into the cytoplasm. IP3 will diffuse into the cytoplasm, where it will bind to IP3 receptors, which are present on the endoplasmic reticulum. There are specific receptors which are linked to calcium channel on the endoplasmic receptors. These specific receptors which are linked to calcium channel, they are IP3 receptors. That's selective for IP3. Once the IP3 binds to its receptor on the endoplasmic reticulum, this will lead to opening of calcium channel, and calcium will rush out of the endoplasmic reticulum, because we know that calcium usually it is cytotoxic. It is usually stored stored into the endoplasmic reticulum. Okay, now IP3 as a second messenger, it will bind to its receptor on the endoplasmic reticulum, opening calcium channel. Calcium will rush out, increasing calcium intracellular. So calcium in this case, what it will do? It will bind with calmodulin, another calmodulin will become active and will form calcium calmodulin complex. Calcium calmodulin complex. Calcium calmodulin complex. This calcium calmodulin complex will activate protein kinases, cellular protein kinases inside the cytoplasm. Cellular protein kinase type C. Protein kinases, cellular protein kinases type C, or BKC, abbreviated as BKC. These protein kinase C, there are 3 different types. The protein kinase C, either they are known as novel protein kinase C, which needs DAG only for their activation. They don't need the calmodulin complex. They need DAG, which we'll talk about in minutes. And there are the classical protein kinase C. Classical protein kinase C, they need both DAG and calcium for their stimulation. They need diacylglycerol and calcium for their stimulation. And there is typical protein kinases which do not need any of these two. Okay, now calcium is released from the intracellular side. Once calcium is released, it will combine with calmodulin, which is a protein present in the cytoplasm. This complex becomes highly active. It will activate other cellular protein kinases. Protein kinase C. Protein kinase C, there are three different types. Novel protein kinase C, which needs only DAG for its activation, or classical protein kinase C, which needs both DAG and calcium. Okay? Now, as you can see here, this is the protein kinase C, which is the classical protein kinase C. This is protein kinase C, a classical protein kinase C. It needs calcium and DAG for its activation. Needs both calcium and DAG. Okay? Once protein kinase C is activated, it will lead to complex cellular actions. Complex cellular actions. Let us take an example. Let us take examples. Once protein kinase C are activated, they will lead to different cellular actions. Very complex, different cellular action. To make it easier for you to understand, let us take an example. Let us take an example. For example, in the smooth muscles of the blood vessels, for example, once calcium is released in the smooth muscle blood vessels, it will activate light chain myosin light chain kinase. Myosin light chain kinase. This myosin light chain kinase is the enzyme which gives energy to myosin to slide over actin. Once actin slides over myosin, this will lead to muscle contraction. This is an example. So myosin light chain kinase is a protein kinase. Protein kinase C. Myosin light chain kinase is a protein kinase C. Myosin light chain kinase, once it is activated by the complex calmodulin, calcium calmodulin, it will be activated. Once it is activated, it will have energy to give it to myosin to slide over actin, and muscle will contract. Other examples many, like it leads to phosphorylation of transcription genes. This is one example. Second example is phosphorylation of transcription factors. These transcription factors, phosphorylation of transcription factor, will lead to, as we know, to cell growth and differentiation, and of course involvement in the oncogenesis. Also, phosphorylation of, for example, of the proton pump, and phosphorylation of proton pump will lead to proton secretion. For example, neurotransmitter release, process of exocytosis as well depends on the presence of intracellular calcium and results in neurotransmitter release. So this is very briefly how Gprotein will activate phospholipase C beta subunit or beta isomer, where agonist binding will activate the Gprotein, and the activated alpha subunit of the Gprotein will activate phospholipase C beta isomer, which will convert phospholipid of the membrane, which is phosphatidyl inositol biphosphate, and convert it into two second messenger. First is diacylglycerol, which stays floating in the cytoplasm, and second IP3. IP3 is a small molecule soluble. It diffuses into the cytoplasm. It binds with specific receptor on the endoplasmic reticulum, opening calcium channel on the endoplasmic reticulum. Calcium will rush out, binding with calmodulin, forming an active complex, calcium colmodulin. Calcium colmodulin can activate different protein kinases C type, like mild chain, myosin light chain kinase, which will result in muscle contraction, or it can phosphorylate different transcription factor, or phosphorylate the proton pump, resulting in acid secretion, and so on. Okay? So this is an example, only an example, very simple example of how G protein can activate phospholipase C beta subunit. So what we have talked about today? We talked about enzyme-linked receptors, and the most important are the tyrosine kinase linked receptors, like insulin, like growth factor, epidermal growth factor, and we have said that these are the site of action of many drugs, especially the hormones. Second, we talked about Gprotein linked receptors and how Gprotein acted as a mediator between a receptor and ion channel, or a receptor and membrane enzyme. We talked about the structure of the receptors linked to Gprotein and how they are formed of single polypeptide chain which crosses the biological membrane 7 times. That's why they are known as 7 transmembrane receptors. We talked about the types of Gprotein, and we said that it is trimeric. It has 3 protein subunits, alpha, beta, and gamma. The alpha can separate from the beta and gamma once they are activated. We talked about the activation of G protein into two active subunits, G alpha subunit and G beta and gamma subunit. We talked about how the G protein can activate ion channels, and we have taken example of muscarinic type 2 receptors which are present on cardiac muscle. And we talked about how Gprotein can activate membrane enzymes, and we have taken the 2 most famous examples, adenylyl cyclase and phospholipase C isomer B, and how they lead to the formation of what are known as second messengers. With second messengers will affect many of cellular actions. So by this we have finished our talk about receptor linked to membrane enzymes and the receptor linked to G proteins, and we have finished talking about different types of receptor. In the next lecture, inshallah, next presentation, we'll talk about what are the different types of ligand that bind to the receptor. So we talked about the types of receptor. Now we'll talk about the drugs that bind to the receptor. What are they? How they differ from each other? So next presentation we'll talk about types of ligands that bind to the receptors. As well, we'll talk about how the receptor are regulated, how the function of the receptor is regulated. This is very important, especially after chronic drug administration. So I have given you 2 references which I have collected the information from. You can refer to for any further reading you want or you'd like. I would like to thank you very much for your attention. If you like the presentation, don't forget to press like and please subscribe to my channel. I hope that you enjoyed my presentation. I hope to see you again in the next presentation. God bless you. Happy New Year to all of you, and I wish you all peace, success, and prosperity. Assalamu alaikum wa rahmatullahi wa barakatuh.